Okay. So, hi everybody. My name is Dorra Hmida. I'm professor in human molecular genetics at the Department of Human Cytogenetics, Molecular Genetics and Reproductive Biology at Farhat Hached University Hospital in Sousse, Tunisia, and in the Faculty of Medicine of the same city. So today we're dealing with triplet repeat expansion disorders. DNA repeats expansions are the underlying pathologies in about 20 to 30 neurodegenerative and neuromuscular disorders in humans. They are classified in two basic groups depending on the location of the relevant triplet repeat in coding or non-coding regions of the genome. Triplet repeat disorders are associated with coding regions, and those ones include Huntington disease, Kennedy disease, DLPLA, and different types of spinocerebellar ataxias. These disorders are also called polyglutamine diseases, since all of them are associated with repeats of CAG triplets, which code for the amino acid glutamine. The pathology of all diseases, all polyglutamine diseases in this group involves pathologies of the nervous system. The non-coding trinucleotide repeat disorders include different disorders, which are Fragile X syndrome, Fragile XE syndrome, Friedreich ataxia, myotonic dystrophy, and some spinocerebellar ataxias. Some of these disorders have been found to be neurodegenerative, such as Fragile X, and while others, like myotonic dystrophies, affect muscle maintenance. The size of the repeat region between individuals varies. It is polymorphic in normal, healthy individuals. Those healthy individuals have variable number of triplet repeats, but there is a threshold beyond which a high number of repeats cause disease. This threshold varies in different triplet repeat expansion disorders. This triplet repeat expansion mutation is also called a dynamic or unstable mutation because as the gene passed from parents to offsprings, the number of triplet repeats increase. In this way, the condition may worsen, so be more severe, and have an earlier onset from generation to generation in the same family. This phenomenon is called genetic anticipation. So genetic anticipation is the increasing severity and earlier onset of the disease as it is inherited through successive generations within the same family. This is well illustrated by this family pedigree, which is specific for myotonic dystrophy, which is a triplet repeat expansion disorder that we will deal with in further slides. So in this pedigree, one can notice that the grandmother, which carries 120 repeats, has developed her first signs at 55 years. She transmitted the expansion to her daughter, and this expansion expanded from 120 repeats to 500 repeats, and the daughter had the first signs at an earlier age at 48 years. She also transmitted this 120 repeat, but expanded to her son, who has 700 repeats, and developed his first signs at 50 years. This daughter, let's come back to this daughter, who transmitted her triplet repeats to her son, so the little son, the grandson, sorry, and this grandson developed the first signs at birth, so had a congenital form of the myotonic dystrophy and carries more than 1,000 repeats. So you can notice that the age of onset becomes earlier through successive generation in the same family, and the severity increases. So what is genetic anticipation? When an individual has a number of repeats in the normal range, it is not expected to expand when passed to the next generation. However, when the number of repeats fall in the pre-mutated range, it is a normal range, but it is an unstable state of the mutation, then the repeats may or may not expand upon transmission to the next generation. Therefore, normal individuals who carry a pre-mutation, so a number of repeats which is in an intermediate range, it's not normal, but it's not fully mutated, so it is an unstable state of the mutation, do not have the condition. This woman, as you can notice, she is healthy, but is at risk of having a child. So she has a boy who has inherited a triplet repeat in the full mutated range. This boy has 500 CGG repeats, and this is a family pedigree of Fragile X syndrome that we will see also later. So this boy has a full mutation range repeat and is affected. So normal individuals who carry pre-mutation do not exhibit a phenotype, but will transmit it to their children. Sometimes the pre-mutation range of the triplet repeat expansion and the full mutation, the affected range, can overlap. In this intermediate area, someone may or may not express symptoms of the disorder. Triplet repeats in the affected range, the full mutated range, often expand when passed to the next generation in the same family. Rarely it will contract to smaller number of repeats, but both situations are possible, and we will see examples through further slides. So this repeat expansion or retraction is postulated to occur via slippage during DNA replication, a number which is not the real one. So triplet repeats in the affected range often expand when passed to the next generation. Rarely it will contract to smaller number of repeats. The likelihood an expansion will occur is based on two facts. It's based on the size of replication, and it's based on whether the triplet repeat is inherited from the mother or from the father. For example, in myotonic dystrophy, triplet repeat is most likely to expand when inherited from the mother, whereas in Huntington disease, triplet repeat is most likely to expand when inherited from the father. So in further slides, we'll see some examples of triplet repeat expansion disorders. Those triplet repeat disorders exhibit autosomal dominant, autosomal recessive, or X-linked inheritance patterns. The more common triplet repeat disorders are autosomal dominant. So the example we are dealing with are Fragile X syndrome, myotonic dystrophy, and Huntington disease. Other diseases will be so studied during the dedicated workshop. So let's begin with Fragile X syndrome. Fragile X syndrome is the most common form of inherited mental retardation. It affects both male and females. It is characterized by moderate mental retardation in affected males, while females exhibit mild mental retardation. The Fragile X syndrome is associated with increased CGG repeats in the FMR1 gene necessary for brain development, which is on X chromosome. The Fragile X relevant repeats is located in the 5' untranslated region of the FMR1 gene on X chromosome. So it is in a non-coding region. It is an X-linked recessive disorder. There is a range of repeat, of CGG repeat, that can cause the disease. The large number of repeats, over 200, causes hypermethylation of the promoter site of the FMR1 gene in the 5' region of the gene and causes a transcription inactivation of this gene. So it is a loss of function. The intermediate range refers to a range of repeat that is normally stable, but in 10 to 30% may change in repeat size when transmitted from a female. An allele in the pre-mutation range, when transmitted from a female, always changes in size, and usually this change is expansion. There is also anticipation, and expansion occurs preferentially in female gametogenesis. This slide illustrates 4 situations where Fragile X is transmitted, whether in mutated state from mutated parent or in pre-mutated state from pre-mutated parent or in a pre-mutated state from a mutated parent. So let's begin with the first situation. The first situation is a mother who carries an X with an expansion over 200 CGG, Fragile X syndrome, and this mother can transmit her X both to her daughter or to her son, and both daughter or son who has her or his mother's X, mutated X, will develop the phenotype. The second situation is a male, a father, with a mutated X and expanded CGG, and this male will transmit his mutation only to his daughters and not to his sons, because his X will be only transmitted to his daughter and his sons will receive only his Y chromosome. And the two daughters who carries her father's X, mutated X, will have a retraction of her father's expansion and will exhibit a pre-mutated phenotype. Third situation, a mother with a pre-mutation, Fragile X pre-mutation, the unstable allele. This mother will transmit her pre-mutated allele to both his daughter and his son, and in both situations, the pre-mutation will expand and will be transformed to a full mutated allele in the daughter and in the son. And the last situation, a pre-mutated father will inherit his pre-mutation to only his daughters, because he will transmit his Y chromosome to his sons, and the daughters will exhibit a pre-mutation and not a mutation, because when transmitted from the father in both situations, the expansion will retract and not expand. So when the pre-mutation is transmitted from the father, it will retract, and when transmitted from the mother, the pre-mutation will expand. So males pass the disease only to their daughters, since they are the ones inheriting the X chromosome from their father, whereas males' offsprings inherit their father's Y chromosome. In contrast, female can pass the disease to both sons and daughters and display less severe phenotype as compared to their male counterparts. The difference in severity of the phenotype are at least in part associated with X chromosome inactivation phenomenon in female. It's a normal, it's physiological phenomenon. Each female cell randomly inactivates one X chromosome. If the inactivated chromosome, X chromosome, contains a defective copy of the FMRP gene, so if the randomly inactivated chromosome contains the X chromosome with the mutation, then the cell will appear normal, because the working X chromosome contains a functional copy of the gene. As a result, female bodies contains a mosaic. So female bodies contains a variable mix of normal and defective cells, leading to a broad range of severity of the symptoms in females. So what is the cellular mechanism leading to Fragile X symptoms? It is a loss of function, the accumulation of trinucleotide repeats, CGG repeats associated with Fragile X results in alteration of the expression of the FMR1 gene, leading to limited or absent protein production in the cell, proposed to lead to disrupted neuronal dysfunction. So this slide showed some clinical features of Fragile X syndrome patient. Males with Fragile X syndrome often have abnormal, as you can notice, abnormal faces, including long faces, large ears, prominent jaw, and macroorchidism, which is not shown here. Affected females usually tend to have milder features than males. Pre-mutation carriers in Fragile X syndrome also exhibit a clinical phenotype. It is specific to Fragile X, and two clinical features are known to be related to pre-mutation carriers. We have the FX-TAS syndrome, which is Fragile X tremor ataxia syndrome, and the Fragile X premature ovarian insufficiency. Both are clinical signs observed in pre-mutated carriers of Fragile X triplet expansion. So now let's move to myotonic dystrophy. Myotonic dystrophy is one of the most common inherited form of muscle disease. It is characterized by muscle weakness and myotonia. Myotonia is a slow relaxation of the muscle after contraction, which progresses slowly over time. It can also affect other organs apart from muscle, such as eyes, heart, and brain. It exhibits variable presentations even within the same family. It is inherited in an autosomal dominant manner. The trinucleotide repeat is a CTG located in the 3' untranslated region of DMPK gene. So it is the second non-coding triplet repeat expansion disorder we have seen. Expansion of CTG beyond 50 copies located in the 3' UTR of the gene causes the disease. There is a normal range, of course, a pre-mutated range, and something which is specific to the myotonic dystrophy is that among the mutated range, we have different groups which refers to different clinical presentation and different age of onsets. So there is also the phenomenon of anticipation. Either parent can transmit the amplified copy. Massive expansions occur only in maternal gametogenesis. So this is the same family pedigree we've seen in previous slides, where you can notice that the grandmother transmitted her expansion, her pathological expansion to her daughter and son, and the daughter transmitted to the granddaughter. While the grandmother exhibits her first signs at 55 years, her daughter exhibits first sign at 48 years, so an earlier age at onset, and the granddaughter at a neonatal age. Besides this earlier age at onset, you can notice that the clinical presentation become more severe with the cataract in the grandmother, myotonia in the daughter, and severe neonatal hypotonia in the granddaughter. GM1, which is myotonic dystrophy, is described so as mild, as is the presentation in the grandmother with only a cataract, classic, as it is in the daughter and the son, which is with muscle myotonia, and congenital, as we describe it in the granddaughter. This clinical variability is based on the severity of the symptoms and the age of onset. The age of onset is earlier and the symptoms are more severe the larger the repeat size becomes. Instability of the repeats is not only described by expansion, which occurs in 10% of cases, but we can also notice some retraction in 3% of cases. Congenital myotonic dystrophy is always due to maternal expansion and never to paternal expansion. So we can see several clinical presentations in myotonic dystrophy patients. So as we already said, it is a multisystemic disorder. So this is a patient faces, which is characteristic of myotonic dystrophy. It is a hatchet face with frontal baldness and ptosis. This is really characteristic of adult myotonic dystrophy patients. So myotonic dystrophy is a multisystemic disease characterized by myotonic muscular dystrophy that may involve the cardiac system at a rate varying between 50 to 80%. Cardiac symptoms are seen usually between 10 and 30 years. The cardiac features include rhythm or conduction disorders that can lead to dilated cardiomyopathy. This cardiac involvement can endanger the vital prognosis. It is the principal cause of early mortality in myotonic dystrophy patients. This cardiac involvement makes myotonic dystrophy one of the rare diseases where pre-symptomatic diagnosis is mandatory for children and minor relatives of myotonic dystrophy patients. This pre-symptomatic diagnosis is indicated from the age of 10 years to detect it precociously a cardiac involvement. So now we'll deal with the last disease, the third and the last one during this presentation, which is Huntington disease. And I've chosen to illustrate Huntington disease, but by its main clinical presentation, which is chorea. So Huntington disease is a rare, progressive, and fatal neurodegenerative disorder. In the early stage, manifestations include subtle changes in coordination, minor chorea, difficulty in mental planning, and often a depressed or irritable mood. In the next stage, chorea becomes more prominent with increasing difficulty with voluntary activity and worsening dysarthria and dysphagia. In late stages of Huntington disease, behavior problems are gradually lessened, motor disability becomes severe, and the individual is often totally dependent, mute, and incontinent. So this disease was first described by George Huntington with a presentation, a clinical description that suggests the autosomal dominant pattern. So it is autosomal dominant, so it can affect both male and females. It has a high penetrance. This means that each time you have the mutation, you'll have the disease, and it is transmitted with a risk of 50% with each pregnancy. The age at onset is usually in the midlife, between 35 and 50 years. It affects 60%, it leads to juvenile forms. The death is expected 10 to 20 years after initial symptoms. It is also responsible of high suicide rates. Huntington disease causative mutation causative triplet repeat expansion is located in the IT15 gene on the long arm of chromosome 4, which encodes the Huntington protein. There is a CAG triplet repeat, and the condition is usually seen when more than 39 CAG repeats are here, leading to the destruction of GABAergic neurons in the caudate nucleus. Huntington disease belongs to the large family of polyglutamine disorders. This expansion of CAG repeats is located in the coding region of the Huntington gene, so in an exon. The 8 CAG repeats in Huntington are translated in a polyglutamine, polyQ disease sequence in the N-terminal region of the Huntington protein. Healthy individuals will carry less than 35 CAG repeats. Usually they carry around 19 CAG, while affected individuals will carry a repeat expansion over 36 CAG with 3 subgroups. Between 36 and 39 CAG, the penetrance is reduced. Over 40 CAG, there is a full penetrance. That means that each time you have more than 40 CAG, you'll have the disease in 100% of cases, and juvenile forms are usually observed with more than 60 CAG. The genetic anticipation, like other triplet repeat expansion disorders, is observed. And in this pedigree, you can notice that a father with a repeat expansion around 32, let me remind you that 32 falls within the high rate of normal expansion triplet repeats. So will transmit this expansion to his daughter and son. This daughter will have 41 CAG and will exhibit a phenotype, will have the Huntington disease. This son also will have 39 CAG and will exhibit a phenotype. He will be affected. And this son will transmit his CAG expansion to his daughter. So the granddaughter will have 47 CAG and will also be affected. What we can notice here is that the age at onset will decrease through generations. The repeat expansion the number of repeats will expand through generations, and I think which is not documented here, but the severity will also increase through generations. The CAG instability in Huntington disease is greater during spermatogenesis. Paternally alleles will expand from 39 to 47, while unstable alleles will retract, in this example, from 41 to 39. The commonest initial presentation in Huntington disease is the chorea, so the loss of motor control and the uninhibited movements. This signs at onset, usually the age of onset, is usually predicted through CAG repeat length. But the CAG repeat length explained only 60% of the predicted age at onset. The remaining 40% are explained by environmental and other genetic factors. So there is place for modifier genes, and as shown by this pedigree, 2 patients with the same CAG repeat length, 39 here, have different age at onset. So to summarize and to move to something which englobes all triplet repeat expansion disorders, why you should refer your triplet repeat expansion disorder patient to genetic counseling. You have to refer them for symptomatic diagnosis, but not only. Patients are also referred for predictive pre-symptomatic diagnosis, prenatal diagnosis, and also for genetic counseling. The predictive testing in triplet repeat expansion disorders are only available for persons who have reached the age of majority, of course, except for myotonic dystrophy because of the cardiac involvement. You need to have with the patient at least 2 appointments before the test for explanations. You have to arrange a follow-up of the patient, and of course, you have to ask the patient for a signed informed consent. In summary, triplet repeat expansion disorders are dynamic expansions of DNA triplet repeats. Normal alleles are polymorphic. Inheritance can be dominant or recessive, autosomal, or X-linked. Pre-symptomatic, symptomatic expansion size varies. The base sequence of the triplet repeat, the location of the repeat also varies. The parent of origin effects on repeat expansion varies. And the stability during meiosis and mitosis also is variable. So your questions are welcome, and thank you.